All right, before we get to the urinary system, just a couple more things to talk about for the digestive system. Technically, it's metabolism, but we cover it with digestive for reasons that I think will be pretty self-explanatory soon enough. All right, so we just talked about how in order to regenerate ATP, get that up all the way, we're going to use phosphorylation of some sort. So that's the general term we use for adding a phosphate group onto something, and in this case specifically to ATP. So substrate level phosphorylation, as I said, is fast but inefficient, and oxidative phosphorylation is efficient but slow, and it does require the presence of oxygen and mitochondria. So what we want to look at is how we use these fuels, these nutrients that we're absorbing to make ATP, and what we do with them when we don't need to use them for that. So for carbohydrates, if we look at catabolism, or breaking them down in order to get energy out of them, pretty much every cell can use glucose, the main monosaccharide, for substrate level phosphorylation. And any cell with mitochondria, which is most of the cells in the body, the major exception is red blood cells. They have to use substrate level because they don't have organelles anymore. If they have mitochondria, they can take glucose and use it for oxidative phosphorylation and get lots of ATP. Glucose is essentially the molecule that the processes of cellular respiration, which use both substrate level and oxidative phosphorylation to make ATP, are based on. Like, everything is really good at using glucose in our body. And glucose is really the only carbohydrate that we use, because the other monosaccharides in our diet, which you don't really need to know, but lactose and fructose, they actually get converted into glucose usually before being used for phosphorylation. So when we talk about carbohydrate catabolism, or breaking it down for ATP, what we're really talking about is glucose catabolism. So if you look up cellular respiration, which is a series of enzymatic events that use phosphorylation to make ATP, you literally get the equation of glucose plus oxygen releases energy and yields carbon dioxide and water. Because carbon dioxide and water are the waste products of oxidative phosphorylation. And a lot of the energy that gets released is in the form of ATP, and a little bit of it is in the form of heat. We can't use the heat as well as we can ATP, but that heat does help us maintain our body temperature. When we look at what we do with glucose when we have more than we need, or how we keep extra around for when we're not getting it straight from the digestive system, we're looking at anabolic processes of carbohydrates. And what we do there is a couple of different things. So we can make new glucose from other organic molecules in a process called gluconeogenesis. So literally, gluco for glucose, neo for new, and genesis for formation. It just means making new sugar. And we can do that out of amino acids, and we can do that out of the glycerol portion of glycerol from triglycerides. There's a couple of different things we can use. We can also store glucose as glycogen. So if you attach a bunch of glucoses together, you get glycogen. And so when we want extra around so that, you know, we don't have to rely on gluconeogenesis or the diet for glucose, we store it. Glycogenesis, glyco, is forming glycogen from glucose. So when we have excess glucose, we can store it like that. Now, both of these happen a bunch in the liver because when it comes to making new glucose, the liver is going to be the major location of that process. And glycogenesis is also something the liver does a lot because it stores a lot. Our muscles do that as well because they tend to keep a certain amount of glycogen stored for future activity. And anywhere where we can make glycogen, we can also break it down to release glucose, which is called glycogenolysis. So that one is actually catabolic, but I just mention it here because I don't know anywhere else to put it. Oh boy, slammed that too hard. Okay, so let's look at how proteins are dealt with by comparison. In order to get into our bloodstream, proteins are broken down all the way into individual amino acids to be absorbed. So when we talk about catabolism, we're looking at the catabolism of amino acids. A lot of amino acids can be used in oxidative phosphorylation, so we can burn them for fuel. Although, you know, we would rather burn glucose and fatty acids. But we'll talk about that in a second. But in order to do that, we have to remove the nitrogen group. So the hallmark of an amino acid is the amine group, which contains nitrogen. But the nitrogen doesn't go in oxidative phosphorylation well. So we have to take it out. And when we remove it, we get this little guy called ammonium. Ammonium is a waste product, and if it builds up, it can become toxic. So we convert the ammonium into a molecule called urea, and the urea gets filtered out of the blood by the kidneys and removed from the body in urine. And we'll talk a little bit more about that as we hit the urinary system next. So typically we use amino acids when we don't have enough glucose or fatty acids to run through for ATP. Or if we have so many extra amino acids that we got to do something with them. Because when we look at protein anabolism, we see that we only make proteins, we only assemble bunches of amino acids together when we need a particular protein. So we don't store amino acids, we have no storage form. So glycogen is the storage form of carbohydrates, but we don't have a storage form for proteins. We either have functional proteins or we have some amino acids that we're going to make ATP out of, or we'll convert them into fat or glucose. We can't keep them around. So when we have excess amino acids, we either use them to make ATP, if that's something we need, and if we don't need them to make ATP, we're going to convert them into glucose or fatty acids for storage. All right, so lipid metabolism is a little bit more complicated, and that's for the same reason that lipid absorption and digestion in the small intestine are a little complicated. So recall that we needed to use bile salts to emulsify the lipid in order to allow enzymes to gain access to them. And then we bundle them up in micelles to allow them to move into the enterocytes. And once they're in the enterocytes, we actually put the fatty acids back together and make triglycerides again. And then that gets packaged with any cholesterol and phospholipids that were absorbed. The enterocytes use these proteins called lipoproteins, which always escort our lipids in the circulatory system. So we call this structure a chylomicron. And these are the ones that actually enter the lacteals of the lymphatic vessels and go through the lymphatic vessel system until they empty into the circulatory system. So chylomicrons look a lot like micelles. They look a lot like anything with phospholipid membranes on the outside, but you'll notice that, and you don't need to recall this, but just pointing it out, that this is a single layer phospholipid rather than a bilayer like in the membrane, and that's because everything in here is hydrophobic. So the tails can just hang out because it's all triglycerides and stuff. And if you had some fat-soluble vitamins in your meal, A, D, K, or E, those would be in there too. And then these are the lipoproteins. They hang out with the phospholipids on the outside because that identifies the chylomicron to the body. And that tells the liver and adipose tissue that there's a bunch of triglycerides in there. So chylomicrons are processed in the liver, or the adipose tissue can pull any of those lipid-soluble products out for storage. The liver is then in charge of making sure that the rest of the body gets triglycerides and cholesterol and phospholipids if they need them. So triglycerides are going to be used for fuel. Cholesterol is used for a variety of things. They're part of the plasma membranes. They're used to make different steroid hormones, remember that? And so the liver will take different lipoproteins and assemble a new ball o lipid, and we call these VLDLs, or very low density lipoproteins. They're very low density because they're really high in triglycerides. So VLDLs are how the liver sends extra triglycerides that it is in possession of to adipose tissue for storage. Like, okay, we don't need them all, so you go take them. So then the adipose takes the triglycerides out without actually removing this VLDL from circulation. So we still have the lipoproteins, we still have some cholesterol in there, and that makes it an LDL, which is a low density lipoprotein. So this has very little triglyceride. It's mostly just cholesterol with lipoproteins and phospholipids on the outside. If a cell needs cholesterol, then they can absorb it. But if we have more of these circulating than we need, then that cholesterol can potentially build up in their blood vessel walls. So when we talk about people having high levels of cholesterol in their blood or their bad cholesterol's up, what we actually mean is that their LDL levels in their blood are high. And the interesting thing about that is the more dietary fats, the more dietary triglycerides you consume, the higher your LDL levels. Whereas the cholesterol levels in your diet actually have very little bearing on your LDL levels. So if you have issues with high cholesterol, it's not about watching your cholesterol in your diet, it's about watching your triglyceride levels. That being said, diet alone doesn't always help because some people do have genetic predispositions to high cholesterol, and there are a lot of other factors. But we do know that because the VLDLs are created to transport triglycerides, and then the LDLs are a result of removing the triglycerides, that's how that is linked together. Now, you may have heard about good cholesterol too. So the other type of lipid transport is called an HDL. HDL means high density lipoprotein, and it's high density because there's no lipid in it yet. It's just a protein shell of lipoproteins that's made by the liver. And it gets sent out so that any other tissues that have extra cholesterol and phospholipids can put them into the HDL for return to the liver. So basically, this is how the liver says, all right, any extras, send it my way. And so this is why we consider this to be good cholesterol, because it's actually removing it from body tissues, and then it will be secreted in the bile. So you'll actually be getting rid of some amount of cholesterol, which is good if you have too much of it. And And that's kind of it in a nutshell. So all of this leads then to what we do with them. This was the best diagram I could find for this. It's really hard to find a nice, simple one that also makes sense, unfortunately. So I tried. But basically what we're seeing is like my cells from the digestive system, and they go to the liver, and then the liver sends the VLDLs out, and then as the fatty acids are removed, we get the LDLs. And this talks about how too many LDLs will end up with bad things happening to your blood vessels. They're supposed to get pulled back into the liver though. And then your liver sends out the HDLs to pick up other stuff, pick up cholesterol and stuff from other places. So that's kind of how that works in a nutshell. All right, so what do we do with these lipids? So we can also use lipids for phosphorylation. Triglycerides are the primary one that we use because phospholipids and cholesterol can be used for a lot of other things, and they have other roles in the body. But triglycerides are literally the energy storage molecule of lipids. So we either store them for the future or we break them down for ATP use. The specific processes that we put triglycerides through are called beta oxidation. This is where we break them down and process the fatty acids, because a triglyceride is just 3 fatty acids strung together. They get chopped up into tiny little bits. We call those bits ketone bodies. And the ketone bodies can either run through the same oxidative phosphorylation steps that glucose runs through, or if the liver is trying to supply glucose to the body and it doesn't have enough stuff to make glucose with, it can send the ketones out instead as an alternative to glucose for cells that need glucose. The opposite of this, so the anabolism portion of this, is lipogenesis. That's what we call it when we make triglycerides for energy storage. And so all the fatty acids that aren't used for oxidative phosphorylation, as well as the glycerols, because that makes up the little part that sticks the fatty acids together, get made into triglycerides and sent to adipose tissue for storage. It's surprisingly easy to do this. In fact, when we have excess glucose, we will typically just reverse the oxidative phosphorylation process at the point where we string them together to make fatty acids instead of making glucose again. So it's super easy for us to take sugars and turn them into fat, which is why having something really high in sugar claimed to be fat-free is silly. What was I just eating today that said that? I thought it was really funny. Oh, it was the applesauce. I was looking at the applesauce, and the applesauce was like, this is a fat-free food. And I was like, yep, that's because you're just apples, but there's also a lot of sugar in there, so potentially that could turn into fat if I eat too much of you. So thanks for that. Anyway, that's lipid anabolism, okay? So lipogenesis, creation of lipid. And of course, adipocytes are really good at this, and the hepatocytes are the other place where this is likely to happen because they're going to be the ones that are cranking out a bunch of extra molecules if the rest of the body needs it. If this sparked something because you've heard of the keto diet and you want to read more about it, here are a couple of decent links about it. We don't have time to go into it. So yeah, there's some good articles. All right. One more thing to talk about, which is the metabolic state. So now that we understand all the different pieces, we're going to put this together and see what happens when we're digesting a meal versus when we're not digesting a meal. All right, so we live in a state of dynamic anabolic catabolic balance. So just like everything else in the body, we're in homeostasis as far as whether we're needing to use stored nutrients to make ATP or whether we're storing nutrients for future energy use. And so we're always going back and forth between those two things, and it's just based on whether or not we're still digesting a meal. It's a little bit different for amino acids. So an amino acid pool would be if we had a storage form of protein, but we don't store amino acids. So when we need more amino acids from our body, our only choice is to actually break down useful protein. How much you need to draw on your nutrient storage depends on when you last ate. So we divide up the body states into an absorptive state when you're digesting a meal and a post-absorptive state when you're not. And this is just kind of showing how all three of these categories of organic molecules can both be converted into polymers anabolically, and how they can be used for phosphorylation, which is what's happening right here, to make ATP. So the absorptive state is also called the fed state, and it's the time it takes you to process a meal and absorb all the nutrients out in your small intestine. It typically lasts about 4 hours after eating, although it does vary based on a lot of factors. But it's basically the amount of time it takes for a meal to move all the way through the small intestine and therefore have all of the nutrients possible absorbed from it. During that time, our anabolic processes exceed our catabolic processes because we have a readily available circulating supply of these nutrients. We've got plenty of glucose, we've got plenty of fatty acids, and plenty of amino acids coming from the gut. So we don't need to draw on our storage. And in fact, we probably have an excess of molecules to what we need. So we're probably actively storing for the future, okay? Which is why our anabolism is exceeding, because we are making glycogen, we're making triglycerides. If we need to make proteins at that point, we're making proteins. And the hormonal regulation for this is going to be those anabolic hormones. In particular, it's going to be insulin, because when your blood sugar levels go up as you absorb a meal, insulin is released because its job is to keep your blood sugar from getting too high. It'll also get released with amino acid levels rising, but that's a whole other story. Insulin is actually aided mostly by the parasympathetic nervous system. So remember that it's in charge of rest and digest. So it's going to be promoting an autonomic state that favors storing your nutrients, being calm and chill and all that stuff. So we have this nice flow chart that kind of shows that. So because it's fed state, we have nutrients being absorbed from the intestine, which is up here, into the blood. And so when the liver is taking all of that in, it's going to be making a lot of glycogen. And the insulin is increasing blood glucose absorption by all the different types of cells. So like the muscle cells take in glucose, they're also going to make some glycogen. The adipose cells will take in glucose and the other nutrients, they're going to make triglycerides. And everybody that needs oxidative phosphorylation is going to be able to take in glucose and use it, because that's what insulin does. It promotes the uptake of glucose. Once we're done absorbing that meal and the small intestine is empty, we now enter the post-absorptive state. This is also called the fasting state because now we don't have nutrients coming from the intestines. And that means that in order to continue to make ATP, we need to tap our body reserves to meet our energy needs. The main goal in the post-absorptive state is maintaining a normal blood glucose level. And that's because there are some tissues that need glucose, that can only use glucose, and they need a steady level of glucose in the blood in order to ensure that it's continuing to diffuse into those cells. The hormone in charge of that is our major catabolic hormone, glucagon. So glucagon is released when blood glucose levels drop below normal. And we also have basically no insulin release. And so glucagon acts on the liver, and the lack of insulin means that most tissues cannot take glucose in. So our glucose is mostly going to come from the liver to supply the body and the blood. The liver is going to break down glycogen with glycogenolysis, and it's going to make new glucose, so gluconeogenesis, from things like amino acids. It can use the glycerol, actually, from triglycerides and other things. And so that's going to maintain our blood glucose. Our skeletal muscle can also break down its glycogen stores to continue to meet its ATP needs. In addition, in response to the glucagon levels, both the liver and the adipose tissue are going to break down triglycerides, so lipolysis, which releases fatty acids into circulation so that it can be used by a variety of cells. Finally, proteins can be broken down by cells so that amino acids can be used for oxidative phosphorylation. So we can have catabolism of cellular proteins. Obviously, we don't want to do too much of that because, we were using those proteins. And so the more of those we remove, the less we have to have functions. So that's obviously more of a last resort situation, and major protein catabolism is reserved for true starvation. In addition to supplying glucose, we also have these glucose-sparing actions. That's what we call your body when there's no insulin present to allow cells to pick up glucose. So really the only types of cells that need glucose are your neurons, so mostly that's in your brain, and your red blood cells, because your red blood cells don't have mitochondria, and for whatever reason, your neurons don't do fatty acids or amino acids. But the other cells in your body do. They're perfectly capable of using fatty acids and amino acids for oxidative phosphorylation to make their ATP. So without the presence of insulin, they can't bring glucose into the cells, and so they use alternative fuel sources instead. This lets our brain and our red blood cells use the glucose that our liver is making sure is still circulating in our blood. Okay, so this is kind of the big thing in the post-absorptive state. And so this is our flow chart of that. It shows all the same tissues, but now instead of bringing glucose into these cells, we're releasing glucose as well as lipids. So adipose is releasing lipid in response to glucagon. Muscle can release glucose from its glycogen stores as well, apparently, and break that down. I always forget that part. I just think its glycogen is for itself, but I don't give it enough credit. And of course, the liver is going to be releasing a lot of glucose. It can potentially release ketone bodies. It can release fatty acids to provide fuel for the rest of the body. And this all gets triggered when the blood sugar drops. That's what triggers the release of glucagon and really puts us into this post-absorptive state. So we can see that the pancreas is kind of our key here. So the exocrine pancreas is critical for digesting our nutrients, and then the endocrine pancreas has these two hormones that are the major players in our metabolic states. And then the liver becomes the major organ that actually carries this stuff out and make sure that the body is staying in homeostasis. So this is a nice little flow with less words, showing the sort of the feedback loops of this. So again, decreasing blood glucose levels will stimulate glucagon release. Glucagon acts on the liver to release glucose and the adipose to release fatty acids. And so then we get this glucose sparing, we get an increase in blood sugar, and that's going to be a negative feedback to shut off the glucagon, and we'll just kind of fluctuate just around our normal blood glucose level until we eat another meal, and then it all goes back to absorptive state and insulin and all that. So we do have those other hormones that are going to have effects too. So for example, hypoglycemia will actually trigger sympathetic activation. So whereas in the absorptive state, it's parasympathetic that's helping. In the post-absorptive state, our sympathetic system can actually help because the epinephrine release will actually essentially back up the actions of the glucagon. So getting the liver, skeletal muscle, and adipose to release glucose and fat. And then other hormones can have an effect on metabolic rate and blood sugar and stuff, but we don't see them acting in that moment-to-moment, between meals, 2 meals kind of a thing. They're going to have more long-term effects. So we're not going to worry about what else they can do. We'll just leave it at that for now. All right, does anybody have any questions before we move on to urinary? All right, let's go to the next one then. We are going to spend today and Monday on urinary system. So we just talked about how we bring a lot of stuff into the body. Now we're going to talk about how we get a lot of stuff out of the body. So we're going to go through the components of the urinary system and touch on all the major anatomy today. And then on Monday, I'm going to cover the physiology of like how we make urine and all the various regulatory and homeostatic mechanisms that the kidneys have, which we've started to touch on a little bit, but now we'll go into a little bit more detail. The urinary system's kind of weird. So as you're going to see as we go through it, the urinary system is basically just the kidneys and then some stuff afterwards. So we're going to touch on the stuff afterwards first and then spend the rest of our time on the kidneys because they really are the system. So we're going to start with the urine and work our way backwards. So urine is the fluid waste product of the urinary system. And I'm sure you're all familiar with the basics of it. It's usually yellow to some extent, unless you've been drinking a lot of water, in which case it's basically clear. Its pH is highly variable. And I'm going to talk about what urine-specific gravity is in a second, but it has quite a range depending on your level of hydration. There can be a few white blood cells in there, but there shouldn't be any red blood cells. So you shouldn't have any blood in your urine. That's not normal. You shouldn't have any sugar in your urine, nor ketones, nor proteins or amino acids. Those things are bad. And the reason why that matters is because we can, of course, test the urine for those things so that if there is some sort of problem, detecting these things in the urine can potentially point us to it, especially when the problems are in the kidneys or the rest of the urinary tract. So the process of testing urine is called urinalysis. If you're curious about that, here's a cool link on how to do it. It kind of explains all that. All right, so USG means urine specific gravity, and it's a measurement of urine density, which sounds weird, but it basically just means how much stuff is dissolved in the urine or in the water. How much of this is water and how much of this is stuff dissolved into it. It's not used as much anymore because now we just measure the thing we actually care about, which is urine osmolarity. But this is still really handy. We just use this little handheld device and like you look through it and it looks like this and it tells you something. So for comparison, the specific gravity of water is 1.000. So there's always going to be something dissolved in the urine, which means that urine has to always be greater than 1.000. And typically it's somewhere between that and like 1.030, okay? And when it's really low and it's closer to water, then you're probably pretty hydrated because you're getting rid of your excess water. And when it's higher, closer to that, then you're probably more dehydrated and you're trying to only get rid of as much urine as is necessary to get rid of the waste products in it. So that means that your urine volume is going to vary depending on a lot of different factors, mostly based around hydration. And we'll address that a little bit more on Monday. So there is a minimum amount of urine that you need to make every day in order to get rid of metabolic wastes, mostly that urea from the amino acid catabolism. And that's 500 milliliters a day. That's half a liter. Off the top of my head, I can't remember what that is in whatever we use here. But normally we're not on the edge of dehydration all the time. Like we're usually drinking enough water. So most people make somewhere between 1 to 2 liters a day on average. We do have terms for abnormal amounts of urine being produced. So if you're not able to make any, it's called anuria. That's bad. It usually means your kidneys aren't working. If you're making some but not enough, we call it oliguria. And if you're making more than you should be, it's called polyuria. So just so you know, we do have some terms for that stuff. If you're going into the medical field, you may encounter them someday. Urine pH is also highly variable. So we can have acidic urine, we can have alkaline urine, we can have neutral urine. A lot of it is based on diet. Essentially, higher protein diets are going to be acidifying. Higher plant diets are going to be more alkaline urine. And we'll talk about how that works again on Monday because that's physiology. All right, so the urine comes out of somewhere. So we're going to trace its path back up and mention how it comes out in the first place. So we divide the urinary system into 2 major sections. The upper urinary tract is the kidneys. They're the ones making the urine. And the lower urinary tract is everything else. So the ureters, which connect the kidneys to the bladder, and then the urethra, which is where the urine comes out. Okay, so their job is transportation of urine, storage of urine, and then voiding is another term we can use for peeing. The kidneys are continuously forming urine, and the ureters will constantly transport it. So the only way to not constantly be peeing a little bit all the time is to store it in the bladder. All right, so we'll start with the urethra, which is the tube for elimination. It's also the only portion of the system that differs between males and females. So we haven't really encountered any significant differences so far because the only major system that varies between men and women is the reproductive system. But the urethra is something that in men is actually shared between the urinary system and the reproductive system. And so it does have an anatomical difference depending on your biological sex. So either way though, the opening to the urethra, so the connection between the bladder and the urethra is at the base or bottom of the bladder. It's in a region called the trigone. I'll show you that in a second. And just like the anus, there are two sphincters to regulate whether or not things are coming out. So regulating urine voiding. So we have an involuntary smooth muscle sphincter, and we have a voluntary skeletal muscle sphincter that is external. Exactly the same setup as for defecation. Here are the urethras. So the female urethra is quite short, averages 4 centimeters, and it's embedded in the vaginal wall. So we'll look at that a little bit more when we get to the reproductive system. But basically, the bladder is here, and the urethra is here, and the opening is right down here. And the reproductive organs are more back here. So they're next to each other, but they're not actually the same thing. And then here is what we just studied. So here is rectum and anus. In men, we actually divide up the urethra into 3 sections based on the tissues that it is running through. So the prostate, which is a male reproductive organ that we'll talk about later, sits right at underneath the bladder at the base. So the urethra runs through it first, so we call that the prostatic urethra. And then the urethra has to exit the body wall. This is where it runs through some skeletal muscle, so they call that the membranous urethra. I don't know why, this makes sense to me, but whatever. And then the rest of it is running through the penis, and that part is called the spongy urethra because the tissue that surrounds it I've blanked on it. Well, anyway, it's got a spongy word in it there, and that'll make more sense when we have the reproductive system. So you can see much longer because it's going to run the length of the penis and open at the tip. And so that also means it's more variable. We'll cover that when we get to reproductive system. All right, so we go up the urethra, we get into the urinary bladder. Now we usually just call it the bladder, but technically it's the urinary bladder, right? Because we also have a gallbladder. So its job is storage. And all of these things, they do have the same 4 layers as we saw in the digestive system. So we're gonna have a mucosa and a submucosa and a muscularis. And most of these guys have adventitia rather than serosa because they are not in the abdominal cavity. Now the mucosal layer here has transitional epithelium, which hopefully I touched on well enough in the tissues lecture, but transitional epithelium is unique to the urinary system because it's really good at stretching and then shrinking back down. The smooth muscle of the bladder is called the detrusor muscle, and that includes the part that is thickened at the opening of the urethra and acts as a sphincter. Now the bladder is really good at storage, so it can go from nearly empty to holding more than 500 mils or milliliters of fluid. And since the minimum amount of urine that you make in a day is about 500 milliliters, that means that I guess if you timed it properly, you'd only have to pee like once or twice a day, but I don't recommend that. Stay hydrated. For the most part, the bladder is retroperitoneal, but as it fills with urine, it sticks into the abdominal cavity. And so when we look at the anatomy of it, we see that the peritoneum actually sits on top of it like a little cap. And so that part has a serosa, and then the rest of it just has adventitia down here. And then the ureters are retroperitoneal as well, and they run up to the retroperitoneal kidneys. And the urethra is not even retroperitoneal, it's just not in the cavity at all, so we don't even worry about it. Okay, so here is the sphincter. It's just thickening of the smooth muscle of the bladder where the urethra opens up. This is the trigone right here, because it just kind of looks like a triangle. And the ureters, the tubes connecting the kidneys to the bladder, they empty right there. So we've got emptying of the ureters right here, the bladder can fill from there, and then the opening to the urethra right here. Make a little triangle. And then this is showing you what the transitional epithelium looks like. So it looks thicker when the bladder is mostly empty, and then it looks thinner as the bladder expands, but it always maintains like a waterproof nature so that urine can't leak out of the bladder walls. All right, since we're at the bladder, we're going to talk really quickly about the function of urination or voiding. The super scientific term is micturition. Don't know why. And just like swallowing and defecation, this is a combination of involuntary and voluntary actions. It's set up very, very similarly to defecation. So every couple hundred mils or so, about 150 mils is when it starts, the bladder is stretched enough that it has these stretch receptors that say, hey, we're getting full, maybe you want to empty. And we call that an urge to void. That's when you're like, oh, I have to pee. Okay, that's just the fancy term for, oh, I have to pee. When we're babies, we pee. Basically, that's what happens. But when we get old enough, we learn to recognize and then override this reflexive action by using our voluntary control of that skeletal muscle external urinary sphincter. If we keep it contracted, the same thing happens in our bladder as happens to the rectum, where the smooth muscle relaxes and it'll keep filling. But every couple hundred mils or so, the stretch receptors will get triggered again and we'll be like, oh, you know, I gotta pee. And the more full the bladder gets, the more the sense of urgency builds, right? You're like, no, I really have to pee. When you get near that 500 mil mark or whatever it is for your particular bladder at these high volumes, you will get to a point where you cannot say no anymore. And the involuntary reflexive relaxation of both the internal and external sphincter will override your conscious control, because it's more important that you don't rupture your bladder by trying to overfill it than, you know, that you don't urinate at an inconvenient time, which is why we shouldn't push these things if we have a choice, because at some point you're gonna pee. It is gonna happen. All right, so that's basically how that works. And the cool thing is that it works at the level of the spinal cord. So it's a spinal reflex, which means that no brain control is required to relax the internal urinary sphincter, relax the external urinary sphincter, and contract the rest of the detrusor muscle, so the smooth muscle within the wall of the bladder, in order for urination to occur. You need brain control if you want to stop the process, but that means that people with spinal cord injuries above the level of mostly the sacral nerves is where these guys are, above that will still urinate without a problem. They just can't control themselves and they can't stop themselves from urinating. But that's better than if you're damaged in the wrong place, you can't urinate and it'll fill and you need help to let it out. Anyway, sorry, back, getting distracted. All right, so just like defecation, you can use your abdominal muscles to aid the urination, but it will happen all by itself without any conscious help. You just have to make the decision to keep your external anal sphincter or external urinary sphincter relaxed, okay? But I'm sure you've all been there, especially when you've waited and you really have to urinate at this point. You want it out as fast as possible. You can consciously push. All right, so that's micturition. The nerves come out down here from your sacral spinal nerves, and that's showing you the innervation to your external urinary sphincter. All right, so the bladder is filled by the ureters. So the ureters are paired tubes, because we have one per kidney and we have two kidneys usually. They're paired tubes connecting the kidneys to the bladder. So their job is to move the urine from the kidney as it's produced into the bladder. And they have smooth muscle that's capable of peristalsis, just like we see in lymphatic vessels and in the digestive system. And so just like we can swallow while upside down, we can also keep filling our bladder while upside down because we're not relying on gravity for the flow of urine. The ureters enter the bladder in a way that creates a one-way valve. So there's no special structure to it, but it comes in at an angle, and the bladder wall actually keeps it closed unless urine is actively coming out. And that means that we cannot build back up. Urine can't go back up the ureters once it enters the bladder, which is a really, really good thing because that would create pressure and damage the kidneys. Hard to find a picture of that, so I gave you this instead. I don't know how helpful that is, but basically, again, here's that trigone, and if we looked at the bladder wall like as a section, we would see that it's like this. So when we have active peristalsis, this is open and squirting urine, and then in between that, it gets covered, so we can never go back, okay? Very efficient of our bodies to set it up that way. And that's going to take us to the ureters, because that's it. They're not the ureters, to the kidneys, because the kidneys are the most important part. They have the most complex anatomy. They do almost all the work of the system. And so we just like, all right, we covered the lower urinary tract. Let's talk about the kidneys. All right, so kidney anatomy. Renal is the term we use to describe kidneys. They are located retroperitoneally within the abdominal space, but not the abdominal cavity. So we talked about that last time, right? They sit just against your last ribs. So your ribs partially protect them. Your vertebrae partially protect them. And you can kind of see where that is. So that's why, you know, getting punched right here really hurts. I assume. I haven't actually experienced that one, and hopefully I never will. All right, so there's no serosa here, but the kidneys are super important. They need to be really well protected. So there's actually a triple layer capsule surrounding the kidneys. So immediately around the kidney itself, essentially its outermost layer, is dense irregular connective tissue forming a fibrous capsule. So that supports its outer shape and helps protect it. And then around that fibrous capsule is what we call the renal fat pad. So literally we have a layer of fat wrapped around the kidney for shock absorption and protection. And then kind of keeping that fat around the kidney is the renal fascia, which is another layer of dense irregular connective tissue, which basically anchors the kidneys into their retroperitoneal position. And then remember that the adrenal glands sit on top of the kidneys like hats, so they're hanging out within all this stuff too. When we look at the kidneys themselves, hopefully you know that they are kidney shaped. We see that we can divide it up into three regions. So the renal cortex is the outermost. Whenever we say cortex, we mean it's the outer layers. And this is going to contain nephrons, which as we're about to find out are super important. Within that, so deeper, is the renal medulla. So this should sound just like the adrenal glands. Cortex on the outside, medulla on the inside. So the medulla is here. It's kind of weird because it's divided up into these little lobes that are shaped like triangles. The urine comes out at the bottom of these lobes. And then the inside of the kidney is what's called the renal pelvis. And the pelvis is essentially a funnel that collects the urine as it gets produced from each of these guys. And then it's continuous with, so it essentially forms the beginning of the ureter. All right. So here is a drawing of that. Renal capsule around the outside. The cortex is this whole outer layer. The medulla is formed into these pyramids, is what we actually call them. And then the blood vessels come in at the same location as does the nerve as the ureter comes out. and they go in between the medullary pyramids to get up to the cortex, okay? So everything comes in and out at this same place, which is called the renal hilum. So it's the only place that anything comes into or out of the kidneys. The rest of it is surrounded by that triple capsule. So both blood and lymphatic vessels, nerves, and ureters. And it's located medially, so towards the middle of each kidney. And as you can see here, that works really well because the blood vessels that the renal arteries and veins come off of are in between the two kidneys. Oh, right, I had another one. All right, so here's just a different diagram showing all the same things. Gives you a better view of the adrenal gland. Give me 1 second. All right. Oh, I don't know what just happened. Okay, we're still going. Okay, so microscopic anatomy. Pretty much the entire kidney is made of nephrons. Nephrons are considered the functional unit of kidneys. So remember that a functional unit is the thing that does the job of the whole organ, or the whole whatever, right? So we learned about sarcomeres for muscles are the functional unit because they do the contracting. We learned about osteons for compact bone. So nephrons are the same basic concept, but way more complicated, unfortunately. Okay, so nephrons have two main parts. There's a renal corpuscle, which is where we filter the blood, and then the renal tubule, which is the part that converts what we filter out of the blood into the urine, or into urine. And they're tiny, so each kidney has at least 1 million nephrons. The renal corpuscles are all located in the cortex. So the cortex is just packed full of 1 million renal corpuscles. And in order to filter the blood, it's going to have capillaries. So the glomerulus is what we call the ball of capillaries where blood is being filtered. Glomerulus just means like a berry or like a mulberry or something, because it looks like a berry. Now, this is one of the locations where instead of those continuous capillaries that we see almost everywhere in the body, we see leakier ones. So they're fenestrated, which means they have slightly larger holes in them within the actual cells so that they're able to filter more stuff out than would be filtered at a normal capillary bed. Surrounding these capillaries are the glomerular capsule. So basically, to capture the fluid being filtered out of the urine, we have to have something around the capillaries. So we've got these ball of capillaries, and around that, we've got a capsule. It's hollow, so there is space in between. That space tends to be filled with the fluid filtering out of the blood, though. And there's two layers of it. Let me get. So again, we have a situation where we've got some of it covering the glomerulus, and then we've got some of it lining or actually forming the layer of the capsule itself. So the parietal layer forms the outside, the visceral layer covers the capillaries, and then the blood is coming in from that side over there. So the blood's coming in from here and actually coming back out from there. So when you form a ball, you need an exit still. And then the rest of the nephron is a tube, and it goes that way. And so this capsule is actually continuous with this tube. Let me just, oh no. That's fine, I have diagrams. Anyway, so here it is again. So we've got ball o capillary glomerulus. Blood comes in, swirls around these capillaries, filtering fluid out through the visceral layer of the capsule, and then the parietal layer forms the outside, and so we're catching this filtrate in between layers, and then it just becomes the tube, and out it goes to turn into urine. All right, the blood, meanwhile, after being filtered, heads back out because the circulatory system is continuous, so it has to go somewhere. And Bowman's is the old name. Oh, glomerular, I do it every time. Bowman's is the old name. Glomerular is what we call it, okay? All right, so corpuscle. In order to properly filter the blood, the glomerulus has to handle really high pressures, and the visceral layer of the capsule helps with the filtration. So by covering these really leaky capillaries, it forms this thing called filtration slits, so that we're filtering the blood without letting proteins out, because we don't want the proteins to come out. We need it to stay in the blood. So the filtration membrane is the actual layers that blood is filtered through into that cavity, that space in the renal corpuscle. And it's composed of those fenestrated endothelial cells of the capillary wall. Of course, there's a basement membrane because it's epithelial tissue. So there's a shared basement membrane that it shares with the podocytes, which are what we call the cells that make up the visceral part of the capsule. Okay, they have these little processes called petty cells, and they kind of fit together to make these filtration slits. So this allows movement of molecules that are fairly small, less than 70 nanometers. You don't need to know the number, but what you need to know is that proteins and cells are bigger than that. So we're keeping proteins in the blood, we're keeping cells in the blood, we're only filtering out the larger stuff, okay? We're just filtering out basically the liquid, so it's going to be the water, and it's going to be everything that's actually dissolved in the water, so solutes. Electrolytes, nutrients, waste products, sometimes hormones, stuff like that. This is what the filtration membrane looks like. So these podocytes really wrap around and interlock around these capillaries. So we just have these very small areas to actually filter out the fluid. And then this is what those fenestrated capillaries look like. They literally have holes in the cells that allow the passage of more stuff than a normal capillary. All right, so again, nephron is the functional unit, and it's made up of two things. So we just talked about that part, okay? And now we've talked about the tube, which is the rest of this stuff. Before I do that, I just want to show you real quick. So here is the artery that supplies the glomerulus, and then it comes back out, and then it makes a capillary bed around the tube part, okay? Showing you that. Tubule is a tiny duct. So tubule just means like small tube, so it's a duct that leads from the glomerular capsule to the tip of the medullary pyramid. So it's just this long weaving tube that makes urine. And we divide the renal tubule into three regions. There's proximal convoluted tubule, we abbreviate PCT. There's the nephron loop, and then there's the distal convoluted tubule. And then the renal tubule ends by joining up with a collecting duct. So the collecting ducts are the next step in the flow of urine. they get urine from multiple tubules that are then aiming at that medullary pyramid. All right, so the proximal convoluted tubule has cuboidal cells that have microvilli. Remember that microvilli are all about increasing surface area, and in this case, it's mostly for reabsorption. That's what we call it when we're removing things from the fluid that is going to become urine. It's not absorption because it was already in our blood and then it filtered out and now it's coming back in again. So it's again, okay? And that's one of those little things. Excuse me. Okay, the cells will also secrete additional waste products that didn't get filtered out in the first place. And these tubes are super, super tiny. So it's literally just the epithelium, basement membrane, here's the capillary. Oh, give me 1 second. Bad. Yeah, so this makes it pretty easy to reabsorb all the stuff we don't want to lose. And we'll talk more about that on Monday. But these cells are very, very active. They have to work really hard to do this job. Okay, so that's the proximal convoluted tubule. They're called convoluted, by the way, because it just means that they like have a very like twisty, turny pathway. So if something is like, you know, like nice and straight, that's the opposite of convoluted, and just means lots of turns. So we usually represent it like that, but that is not very twisty. It's more than that. Okay, the next step is the loop. It's called a loop because literally goes one way and then loops back the other way. So because it's going one way and then turning, we have a descending limb that moves away from the rest of the nephron. This is moving deeper into the medulla. So notice that we have a dividing line here, cortex up here, medulla down here. So it loops down into the medulla and then back up again with the ascending loop and connects up to the next part of the tubule. It used to be called the loop of Henle. We're trying to move away from that and just call it the nephron loop. But that's why these guys have extra names. Okay, so you can see that the majority of the nephron is in the cortex, and then the loop is in the medulla, and these collecting ducts do descend into the medulla as well. So most of the medulla is collecting ducts and loops of Henle. Okay, the collecting ducts, okay, so the distal convoluted tubules, we'll talk a little bit more about their physiology later. They're, I don't know why, but they never get a special thing for the anatomy. They work with the collecting ducts. So even though the collecting ducts aren't part of the nephrons, because they're the next step, they still have an important role in urine production. And that's because they have water channels called aquaporins that can be inserted into the tube to increase water reabsorption when we need to retain water, like when we shouldn't be peeing it out. So we get more water channels when antidiuretic hormone says we need to retain the water. So remember that antidiuretic hormone is released when we don't want to lose extra water in the urine. You put more water channels in, we reabsorb water, so the collecting ducts still have a role, they're not just a tube. So we typically deal with the distal convoluted tubule and the collecting ducts together because most of their job is determining how much water actually goes out into the urine. And we'll tackle that again in more detail on Monday because that'll be the physiology side. All right, one more thing about nephrons. There are two types. So most of the nephrons in your kidney, 85% are cortical, which means that almost the entirety of it is in the cortex and the loop is very short. It really doesn't go very far into the medulla. But the other 15% have really long loops that go way deep into the medulla. And the nephron itself typically sits closer to the medulla than the other guys. They sit higher up in the cortex. So when things are next to something, like these are next to the medulla, we can use the term juxta to describe them. So we call them juxtamedullary nephrons because the majority of the nephron is closer to the medulla and the loops are really, really long. And this is super important for us to make concentrated urine, so urine with as little water in it as possible. So we'll tackle how that works in a little bit of detail on Monday, but this is the other big thing about nephrons, is that there are two kinds. So the blood supply to these guys is critical to understanding urine formation as well, because anything that we filter out of the blood is going to be coming by way of these arterioles to the glomerulus. So the afferent arteriole is sending blood through the glomerulus, and then the blood comes back out by way of the efferent arteriole. So hopefully you remember when we talked about like the nervous system, so the sensory stuff was like the afferent pathways, because afferent is sending things into something. And then like effectors are like the muscles and things that do stuff. So they were getting information from the efferent pathways, because efferent stuff means coming back out. So the afferent arteriole goes into the glomerulus, and then the efferent arteriole is draining the glomerulus. So the blood goes in, the blood comes back out. So then the efferent arteriole is going to become the paratubular capillaries that wrap around the tubules. And so that's like that little picture I showed you with the proximal convoluted tubule. That's the capillary that's just on the other side of the basement membrane. So the paratubular capillary is going to be receiving whatever the tubule reabsorbs from the fluid that we don't want to become urine. The juxtaglomerular tubules, because those loops are so very long, they have a specialized paratubular capillary called the vasa recta. Vasa just means vessel, and recta means straight, because it follows the loop and there's these crosses that are just straight across, which is a little different from the way that the normal cortical paratubular capillaries work. I put another, I did not. Okay. One more big anatomical point is a major sensor that allows the kidneys to monitor the composition of the fluid that is becoming urine. So the juxtaglomerular complex, which is called the JGC or the JGA because some people call it apparatus, is made up of a couple of different things from different parts of the nephron. So the ascending limb of the nephron loop, right as it becomes the distal convoluted tubule, actually sits against the afferent arteriole as it becomes the glomerulus. So the very beginning of the nephron is actually touching one of the parts that's very near the end of the nephron. And so this part of the ascending limb monitors the sodium levels of the fluid inside of it. And that allows the kidneys to regulate how much blood they're filtering, as well as help monitor overall systemic blood pressure. And we'll talk more about this stuff all on Monday again, but I want to just give you a little bit of it because we've talked about it before. If sodium levels or overall fluid levels get too low in the nephron loop, the kidneys will release renin. They will also change the diameter of the afferent arteriole to regulate blood flow into the glomerulus, which will increase the amount of blood being filtered or decrease it. We'll deal with it later. Okay, so renin hopefully sounds familiar because I'll come right back to this. Renin is the enzyme that activates angiotensin as it circulates in the blood. And angiotensin causes vasoconstriction to increase blood pressure, and it releases aldosterone from the adrenal cortex and antidiuretic hormone from the pituitary gland in order to promote water and sodium retention in the kidneys. Okay, so it starts with renin release from this juxtaglomerular apparatus or complex. And it's because as this nephron loop is coming back up, it sits against this afferent arteriole. And so this spot allows for pretty intense monitoring of the composition of fluid as it's almost urine. Okay, so we'll talk about that more as we work our way through the physiology, but it's good to kind of start thinking about it first. Okay, and this is actually a picture of a microscopic slide that's showing the actual structure. It's kind of cool. So we're going to go over this again on Monday, okay? But I wanted to get you started thinking about it. Anybody have any questions? All right. Keep your eyes out for worksheets for these guys. I will be working on them, and yeah, we'll do urinary system physiology on Monday. Now there's a question. Okay. Oh, thank you. You have a good week too.